introduction. It's really awesome to be back. Not only to have gimme coffee, but also, just seeing people so intellectually engaged. I mean, we have that at all institutions, but I forgot how intense and intellectually engaged everybody is here, and that's really been a real pleasure. And seeing people at the cutting edge, students, postdocs, faculty, etc. So Mike said something really important, and one of the things I really saw at Cornell was I'm gonna point out Mark Sorrells, because Mark Sorrells is one of the few people who first sort of introduced us to this idea of having a breeding program and then using technology to supplement the breeding program through his classes and through his own work. And so that's really what I try and do. So I do have an applied field breeding program, but I try and ask some interesting questions as well. So I'm going to tell you a little bit about, are both slides not going to change?
That one's slow. Yeah.
Okay. I'm going to tell you a little bit about the applied field breeding aspect, because I think as I was a Cornell graduate student, I didn't get to hear a lot about that aspect. I always got to hear cutting edge science, but it was always neat to hear some sort of field breeding stuff and some practical aspects. So I'm going to start off the talk with that, and then I'm going to end with a really neat study we just did in recombination, which I think is a little more basic and interesting on the science side. So maize in Texas is very different than maize in the rest of the country. You can see from this picture right here of where maize is produced, it's all the I states, the Midwestern states. But it's not that maize is unimportant. We're talking about $1 to $1.5 billion per year in farm gate value. That's divided roughly in half. The high plains produces much higher yield, on average about 204 bushels an acre, compared to my area, which we call the rest of Texas. It's not very descriptive, I know, but it really is the rest of Texas versus the high plains. And that area is much lower yield.
Is there anyone here that can fix this? Oh, God.
So this rest of Texas area is lower yielding, mainly because we use rain-fed. Okay, so it's more of a dryland type of system as opposed to the highly irrigated system. Now, one of the things I'm going to show you is how the private sector is not really meeting the needs of our growers. And so the justification they use is there's just not enough value there. But a very conservative estimate of 2 million acres times 3 acres per bag of seed, which is really conservative, $150 per bag of seed when really it's probably closer to 300, means that there's $121 million of seed sales every year. Okay, so there's clearly a lot of value there in the seed business. Now, one of the things that's really interesting about Texas is how much different our yield curve has been from the rest of the United States. So what I'm showing you here in black is the US average yield in maize, and of course that's gone up. Here's the Texas average yield. All of a sudden those high plains came online with their irrigation and mining the Ogallala, and then right about this point, 1995, we see this separation between these two, the US average and the Texas average. And the dryland acres have definitely stayed flat. The irrigated acres look like they've gone up. That can be a little bit misleading though. Okay? So we have a little bit of a challenge. So my student, Ivan Barrero, who's now a breeder at AgriLion, conducted this study, and he collapsed a bunch of different data sources to do a meta-analysis. This component here was actually based on our corn performance trials. So there's about 14,000 observations that went into this data set, most of it of elite commercial material that they were currently selling to our growers. And one of the things I'm not going to show you here, I'm just going to tell you about, is we basically found there was zero yield gain in Texas due to genetics. Okay, and some of the most recent cultivars they've been selling have actually been lower yielding than stuff they've sold in the past, which is quite shocking. But one of the other things I wanted to show you is how strong our correlation in the rest of Texas is between grain yield and plant height. Now, we don't see that same correlation in the high plains. It's still significant, but it's not as substantial. And so Winwei Zhu is the breeder up in the high plains. So we have a little bit of a different environment here. He's high yielding, I'm low yielding. For us, this plant height and also the moisture, which corresponds to maturity, is really important. And what that has to do with, I think, is adaptation. Adaptation to stress and adaptation to the growing season. So this is the USDA plant hardiness zone map. I think most people have seen this. Right? And we have about 20 or some zones across the United States from north to south. This here is the corn maturity map. And you'll see the only place that the corn maturity map really matches up to the hardiness zones is through a few places around the I states. Okay? The rest of the entire South is just considered 120-day corn. So clearly there's a little bit of a bias here. Now when we go and look at where the private companies are breeding, they're mainly breeding in the Midwest. So that creates a little bit of this challenge. There's really only one company in Louisiana that has a breeding station, and Terrell Seeds, and they've really been making a lot of gains because they're breeding in that environment. So one of the things we found from our meta-analysis and from our data is that there's this idea that you can breed the best material in the best environment, and it's somehow going to translate to the poorest environments. And so I think what I've been able to show and to see is that that's just completely not true. And it's really interesting that soybeans, on the other hand, actually their maturity maps correspond a little better throughout the entire United States. So maize has this huge bias just towards the Midwest and against the entire South in terms of specificity of environments. So I do run an applied breeding program. I'm going to show you a little bit more on that in just a minute. One thing I wanted to show you though is why commercial material might do so poorly. So this is our best commercial tester, LH195. It's a Holden's line. And you can see, hopefully in the back there, how small and shriveled up some of those kernels are, covered in fusarium, bad pollinations, and this was an open pollinated set of inbreds. Okay? The inbreds just don't like it in Texas. It's too hot, it's too dry, it's too humid, it's too hot at night. This is our, I think, our game-changing line, TX777. It was derived from some Bolivian material, and it even has some problems as an inbred line. But once you combine those, I think everybody's seen pictures like this before showing the two inbreds and the hybrids. We can obviously recover some hybrids that are quite strong. So a lot of our corn looks quite different than anything else you're going to see in the Midwest. So I'm only going to show one table of yield data because, again, I know most people here aren't necessarily interested in the full breeding program. But we usually use about 12 commercial checks through all of our trials. And so taking the commercial check mean was 128 bushels an acre in 2013. And we had plain lines, all the ones in white are our lines combined with commercial testers. And we use commercial testers because commercial testers are the ones that are going to have the transgenes. So my growers will not be growing corn unless it has Roundup Ready in it. They're just not interested in fighting the weeds that we have in Texas. And then you can see individual commercial hybrids can occasionally outperform, even our best material. But which commercial hybrids these are change from year to year. Okay, so our material is definitely equivalent to or exceeding, in the case of TX777, the commercial material. Our material is different because my predecessor, Javier Bertrand, came out of CIMMYT in Mexico. And he came with this idea that he was going to bring exotic material and adapt it to the southern United States. And so a lot of the material came from CIMMYT. A lot of it came from places like Bolivia and Argentina. And then he combined that with the material in the Midwest that we know has the great yield. And he did that to avoid things like aflatoxin and problems with maturity. And so that created its own unique set of traits and of germplasm. So I recently had a graduate student, Skylar Smith, who Elliot Hefner was kind enough to do some in-kind support with Pioneer on genotyping. And we asked this question. So we've created all this unique material. How is it different from what's publicly available? And so for those of you who work in the maize world, this red section here, and these colors were figured out by structure, and then this is plotted in a principal component analysis. The red lines here are all stiff stalks. LH195 is one of our key testers. B73 is right here, so it's the most extreme of the stiff stalks. Then we have the non-stiff stalks here in pink. And then we have the iodines here in blue, the B73 and Mo17 hybrid, and here's our Texas A&M material and a few XPDPs. So we've generally been shifting our material towards this axis as our material gets better and better. One of the unique things that we also noticed in this principal component analysis is this axis separates stiff stalks from non-stiff stalks. This axis seems to separate those things that can grow in Texas from those that can't. So these things up here, actually, I can't even get seed off of. There's just no seed. We can make 100 pollinations and get 2 or 3, rotten seed. And the stuff down here tends to do much better, including B104 and TX714. So that was an interesting observation to make. And a good use of molecular markers to quantify the genetic diversity in our program. So I think this is the last thing I'll really show on our breeding program. So this is an example of my field maps. They're fairly insane, but we have a lot of different tests. We do a lot of collaborative work with people. Anybody who wants to grow a trial, I'm more than happy to help support if, as long as we have the resources to do so. In general, I have about 7 acres of breeding nursery, and the main thing is yellow corn for our Texas growers, but we also have interest in white and colored corn, and then a lot of different genetics projects. Most of my graduate students work on various genetics projects, as I'm going to talk about briefly a little bit later. We have a few isolation blocks, and an isolation block, for those who don't know, is a really cool way that we create hybrid seed, which otherwise is quite a bit of labor for us. So we just go out and we plant one common pollinator, and then we tassel everything else, and we get a lot of seed, hybrid seed across our various varieties. For our summer yield trials, the way we've set this up is we generally will test a 2-row plot in either 1 rep or 2 reps. Seed is always the limiting factor. If that does well in 1 year, then the next year we'll try and bump that up to maybe 2 or 3 environments, maybe 3 reps, and gradually we increase that as we go, the more and more confidence we have. We don't really start taking notes till after the first year, because things like height and flowering time, they're useful, but they're not that useful that we can afford to do that on 20 acres of yield trial plots. We do a lot of late planting and limited irrigation to induce that stress, because that's the problem our growers face. They face a problem with high heat, high nighttime temperatures, and drought, and you don't get those unless you plant late. We also try and plant early to maximize those things that can yield really well in an ideal environment. We combine harvest all of this, and then we look at the yield data and we make our decisions based on what yields best. And we can do that because my predecessor actually selected on a lot of other traits beforehand. So we've enriched this material, and now we're in the 3rd generation of its own unique sort of heterotic group in this material. We often find row column effects are significant. And the reason why in our environment is because we irrigate through a furrow. So when water runs down a furrow, it tends to stop in the middle, and the things in the back get irrigated the least, unless you get flooding, and the things in the front tend to get irrigated the most. And so those range effects as you go through the field are usually very significant, so we fit that in all models. Also, some rows, the students don't necessarily know that notice that there's no water flowing down them, or somebody forgets to fertilize them. So row column effects are really important. For those of you who don't already fit those in your model, they can really reduce that error variation and allow you to explain a lot more of the genetics and account for that field spatial variation. So everything's randomized complete block, and then fit row column effects post-hoc. And that's just because we have so many trials, it's hard to design the optimal place to put it in the field. So just to give you an idea on the breeding program and what it's like in Texas, so we've already planted. We start planning our yield trials. We start preparing them in January. We start planting them in February down in South Texas. Plant through early March and into April in College Station, and then throughout the rest of the state. We're going to be doing agronomics and field notes for the next few months. We pollinate in June usually. We inoculate various things around the same time. We harvest in July, August, and then we got to turn that material around pretty quick because we planted our winter nursery in South Texas about August 15th or so, and that's about a 6-hour drive. And so for that, we pollinate in October and harvest in December. So we have this really long season corn in the South, and because of that, we can only get 2 seasons a year, but we always have something in the field, except for the end of December and January. And the only time we really have downtime is November. So it makes it really hard to do a lot of the, the basic science research that sometimes we'd like to do, because it takes so long to prepare all these trials, look at your database, what are you going to plant the next year, and make those decisions. And you got to do it very quickly. There's not a lot of time for thinking, or we make mistakes a lot, put it that way. So, why do we need public breeding programs in corn, and especially in my region and all the regions where the companies aren't? The companies are not targeting these extreme environments. They're not targeting Texas, they're not necessarily targeting New York, or North Dakota, or Canada, and that's where you see the strongest public programs trying to develop applied corn cultivars. And I think there's value in that. There's value in that, especially with changing climate, because the companies aren't necessarily thinking about how are we going to address corn needs 10 years or 20 years from now. And it's that public material in these extreme environments that's really going to help with that. We do a lot of unique traits, and I'm going to just talk about a couple of these today. I'm going to show 2 slides on perennial corn, I think. We do a lot of graduate student training, and most of our students go to industry, unlike most of my cohort at Cornell, who almost all went to the public sector. I think all but one student at A&M since I've been there has gone to the private sector. So it's a very different type of environment. But we're really strong in field activities. I have the smallest of all the breeding programs at A&M. The sorghum breeder is much larger, the cotton breeder is much larger, the wheat breeder is much larger. So very, very hands-on and field-based breeding. I'm really interested in new breeding methods. So what ways can we use to improve the breeding process? I'm going to show you a couple slides on that and tell you a little story on increasing effective recombination. And then long-term high-risk research. I mean, that's the reason that I'm in the public sector, is that so that I can do long-term high-risk research that isn't necessarily something perceived as a need right this moment. I'm going to tell you a little bit about aflatoxin. And I have to do this because that's why my position exists, and it's such an important problem, but it's not one you really experience here. So there's this fungus called Aspergillus flavus. In Texas, it usually affects our ears pre-harvest, which means it comes in generally after flowering, or slightly after flowering, really grows in the ear during dry down. And it's federally regulated at 20 parts per billion. So it's one of the most potent toxins that you can think of. Above 500 parts per billion, you have to destroy the corn. So in Texas, what happens is the growers pay this burden. They have to destroy their corn. But in the developing world where this is a problem, it's really the consumers who pay the price. Because if there's not an FDA there to regulate your food supply, and you're eating contaminated corn, you can definitely either have chronic or acute effects. So in Kenya, it was clear 125 people died eating 2,000 part per billion corn. In some parts of Asia, and San Antonio even, where that's combined with hepatitis or alcoholism, you get a lot of liver cancer. And that's more of a chronic effect. And in the US, sometimes these things slip through the regulatory agencies for animal feed. So dogs and turkeys have been killed a lot. But it's really hard to tie numbers to how much the problem of aflatoxin affects human health and affects living standards in other countries. So Benin, it was tied to a 1.7 centimeter decrease in height because these children were consuming high levels of aflatoxin contaminated corn. And I'm showing you a few examples of sporulation here. Here's a single kernel, here's something that a worm brought in, and here's an entire ear. And those are really fun to open. You open those up, you just get a cloud of toxic spores in your face. But the scarier thing is it's these clean kernels that may be the ones that are 2,000 parts per billion. It may be these ones are very high as well, but just because it shows sporulation or doesn't show sporulation doesn't mean it's clean. So we've been able to tie numbers between 14 and 250 million to the loss in US growers to aflatoxin, and so it's an important problem. How do we breed for it? Well, I like to tell people that the hardest thing to breed for is yield, right? Well, the harder thing to breed for is drought tolerance, which is kind of a hazy topic. And then harder than that is aflatoxin resistance. And the reason why is because there's so many confounding factors. Maturity is a huge confounding factor. Husk coverage. So in the Midwest, we've been breeding corn to open its husk up and dry. In Texas, that's the worst things, because if you have these ears standing up and then you get rain, it's going to put those spores right in there and create that perfect environment for contamination. Kernel hardness. So hard kernels tend to resist the fungus, but cattle feeders really want soft kernels. Earworm resistance, I think we're all on board with that. Drought tolerance. When we get stressed, we get aflatoxin. And so we have to do all that on top of this question of yield, which we know has a lot of different component traits. So as a quantitative geneticist, this is really exciting, right? Lots of different traits that contribute to this phenomenon. But it's also a little scary because oftentimes things we map as aflatoxin resistance may actually just be a flowering time gene. So we got to measure a lot of traits to make sure we're actually after the right thing. So have we been successful? I think we have. I and a number of my colleagues have released aflatoxin resistant, I probably shouldn't call that, decreased susceptibility in aflatoxin cultivars. So our hope is that these can be widely adapted and will decrease our loss for producers here in the US and for consumers throughout the world. I'm just going to tell you one short story on GWAS. And the original goal of this study was actually to try and use genome-wide association mapping to map aflatoxin. And that part of it, I would say, is kind of a failure. But the way we design our experiments, because all of them are directed by graduate students, is so that if they fail at one thing, they're going to at least be able to succeed, write their publications, and graduate. That's one of the major goals of graduate student training, I think. And so what we did in this case was we had a panel of 357 diverse lines. So it was the same panel that's used at Cornell, plus a panel that Marilyn Warburton, Paul Williams, and Gary Wyndham put together for the southern locations. And we actually crossed it to 2 isogenic inbred lines. TX714, which is 97% identical to B73, with 2 lipoxygenase mutant knockouts. One for lipoxygenase 4 and one for lipoxygenase 5. That was predicated on a hypothesis that LOX5 helped with drought tolerance, and LOX4 helped with drought tolerance, and LOX5 helped with aflatoxin resistance. But we ended up not being able to find any effect from those 2 genes. So the only thing I'm going to really tell you about is that we test crossed in these hybrids, and we measured yield and height, flowering time, and all the standard traits that a graduate student would measure. And this was part of Gerald De La Fuente's master's thesis and Yvonne Barrero's PhD thesis. Or dissertation. We found these really cool SNPs for yield. Since these were test cross hybrids rather than inbreds, those SNPs for yield, we think, would actually potentially be valuable for a grower. And these yield effects were quite large. 3 to 8 bushels an acre, 5 to 7 bushels an acre, 3 to 5 bushels an acre, and those seem to be additive, so we can pyramid all of those. Okay? A couple of those corresponded to plant height. Now remember what I showed you before with the commercial varieties. Plant height is closely associated with yield. So that actually made us feel a little bit better about these, that multiple traits picked up at least 2 of these SNPs. And we did find some for aflatoxin resistance, but they had such marginal significance, I don't think they were really even worth reporting. Now you guys have seen a lot of GWAS studies. The thing I wanted to share with you is what we've decided to use as an approach to move beyond the GWAS study. Okay? And in this case, you know, we thought, we listed out, the graduate students and I sort of brainstormed, what are all the different things we can do? Well, of course you want to confirm with other studies, but nobody else grows things in Texas. Nobody else grows test cross hybrids generally for GWAS. So that didn't really work. Could we develop isogenic lines? Well, we thought about the labor and time involved in that. And the biggest thing that's scary to me is, what if you put that on the wrong genetic background? So we see huge context dependency in QTL. I think that's been very well demonstrated across crops, and especially in maize now. So if you put it in the wrong genetic background, you may not see any effect from that SNP. Okay? So we thought, well, that's really risky. So what we actually decided to do was develop biparental populations out of our maize breeding program, or other public inbred lines, or XPVPs, we thought would be useful in our breeding program. So even if we fail at confirming these SNPs, we still have some good breeding material that's an outcome of this study. Okay? And the thing that was that allowed us to do that was this cool resource that was generated here and throughout the United States of genotyping the gene bank. So once we could go into the gene bank and look at the variation that was there, we could say, oh, that'd be a good inbred line that we already use that's segregating for this SNP. So the way we've chosen to do this is we got the resolution from GWAS. We're close to the gene if it exists, right? Or genes or functional polymorphism. We don't need any better resolution. What we need is confirmation. And I think biparental crosses might be the best way to do that for my program. So Yuan Chen is a PhD student who is leading up this project. And so we have 3 populations we developed between 110 and 239 individuals that segregate for these various SNPs. And so far we've only had a winter nursery to phenotype these in, but we found significance for all 3 SNPs across either 1 or more populations for plant height, which made us feel like there might be something there. Now this is in inbred lines, it's in an off-season nursery, and it's not in hybrids, which is where how GWAS SNPs were originally discovered. But I'm really excited about this. I'm looking forward. This is our big yield trial year, so we probably got 10 acres alone devoted to this project. I mean, it's massive in the amount of test crosses that we made of it. If those SNPs work, that could be really useful to growers. Now, one of the other things that I'm really interested in is, again, diversity and recycling that diversity and multiple parent populations, making synthetics, making new sources that we can select inbred lines out of. And so what we did is we took those lines that look so good from the GWAS study for aflatoxin, Okay, there were 2 GWAS studies, I'm not discussing the other one. And we used those sources in multiple parent cross populations. Now, what we're doing this year is we've sent these off to various investigators to self-inbred lines out of it. So we can see and test the hypothesis that these things may actually have broad spectrum aflatoxin resistance. We hopefully can select some high yielding stuff out of this. And the cool thing we're going to do is we're going to select phenotypically for yield and using markers and see which one does a better job of transferring the aflatoxin resistance to the progeny. So we have markers for a lot of these things from various studies. We're going to try and use them now. We're going to try and apply that science into something that can be used by a grower. The other thing we're trying on a similar aspect is to integrate these breeding gains with other things we know control aflatoxin. So recently there's been atoxigenic strains, and what that means is the Aspergillus flavus has been knocked out for aflatoxin. We can't produce aflatoxin because of some gene in the pathway is non-functional. And so growers are using this to decrease the amount of aflatoxin. However, when you open up these ears, they're still green and moldy. Okay? It gets, it's like I like to say, it's at the spirit of the law, but or it's at the letter of the law, but not the spirit of the law. You still get really moldy corn. But we're testing those atoxigenic controls with our resistant lines, with various irrigation treatments, and with various levels of transgenes. So we have isogenic commercial hybrids for 1BT or 2BT or for Viptera. So we're testing all of these things together to see if they can actually make an additive difference or if there's any type of weird effects in there, epistatic type effects. Taking a bunch of traits, so our results really shocked us. So this is from the first year, this is just preliminary data, and this year is another study. But we found multiple cases where the irrigated corn had more aflatoxin than the dryland corn. That's sort of one thing we would expect. And these are all hybrids. Each group of lines here is a hybrid with one of our Texas A&M breeding lines with a different commercial tester. So on this tester, irrigated had more aflatoxin than dryland. In this case, dryland had more aflatoxin than irrigated. That's something we were not completely surprised to see. There was no significant irrigation effect in this test cross hybrid, but a huge benefit from the atoxigenic strain. But what really blew us away was there were a few cases where we added the atoxigenic strain to that hybrid and it actually increased aflatoxin. So that suggests that you can't just combine these various control strategies without actually doing the testing to confirm that it's going to work. I mean, if I was a grower and I added the atoxigenic strain and it costed $10 an acre and it increased my aflatoxin, I would not be very happy. Okay, so we really need to test these things as a system rather than just in isolation. So the last, most of the last of the talk here, I'm going to tell you about a new study that we're just kind of wrapping up the first component of it. With Adam Mahan, a very hardworking and industrious graduate student who's finishing up this semester, also to go work for AgriLion. And Dr. Patricia Klein, who does a lot of the genotyping, GDS type of techniques in her lab. She's definitely an expert in that area. But what we wanted to look at is the effect of recombination, okay, and it's specifically effective recombination. And Adam had already had some experience working on major color genes, and so that was sort of the impetus of this project. So I want to start off with a hypothesis on this, which is I think in plant breeding and in genetics that effective recombination is our limiting factor. Okay? So it's a hypothesis, it's a testable hypothesis. It's important to remind everybody that effective recombination is what you can detect using markers. Right? It's going to be less than the total meiotic crossovers, because you can't detect everything with markers necessarily. And that's going to be less than the double-stranded breaks, because a lot of those resolve as non-crossover events. But the cool thing about effective recombination is we can measure that with molecular markers. So how do we phenotype recombination? Well, you can do it with a cytology, but it's fairly laborious. If we can use markers, we can make maps fairly quickly on progeny. The reason I think this is important is if we have a little integration like that, that's great in a wide cross. Right? There's not going to be much linkage drag with those. And if we're trying to map something, that's how we're going to get to near gene resolution. We don't want to be identifying whole chromosome arms in linkage mapping. We want to be identifying something close to a gene. So I did some simulations a number of years ago using the NAM data to figure out what the recombination rate is in NAM, and then apply it to different type of breeding scenarios. So what I'm showing you here is the mean effective recombination events after that type of cross. So a biparental cross to F2, the mean number of events is 1.83 per chromosome, okay, per individual. If we go to F8 RILs, that number doubles, as we would expect based on theory. Now if we go to a four-way cross or a multi-parent cross, we can get another huge increase. And what it also does is increases our number of effective recombination events per generation. Right? As a breeder, I want to make the most progress per generation. So the most progress is coming from biparentals to F2, but dihybrid crosses, if we need RILs, seem to be quite effective. Now we can also take a IBM strategy or intermated strategy. So we can intermate for 4 generations, and that is an effective recombination rate per generation of 0.81. And if we combine a 4-way cross with intermating, we can really bump that number up. Okay? So that's the hypothesis based on simulation. So we designed a population to test this. And so these are 4 breeding lines from the program, an oleic version of E73, 2 blue corns, TX903 and 906, and an aflatoxin-resistant corn out of the program. We made the hybrids and self-out recombinant inbred lines. So we have 140 recombinant inbred lines from this 2-way, 140 recombinant inbred lines from this way, or 134. We also made the 4-way cross, self-out lines from that. We did 1 generation of intermating, 228, 2 generations, and 3 generations of intermating, self to essentially RILs. We have 527 inbred lines. So it's a huge population of over 1,200 inbred lines that we're using to sort of test these hypotheses. Now, how many markers do we need? So based on my simulations in NAM, there were 3.56 events that I expect to be detected with the average of 84 polymorphic markers. NAM actually detected 3.58 by my calculations. With 1,000 markers, we figured we'd detect 0.5 more effective recombination events. And critically, those are going to be those small effective recombination events that we're most interested in for mapping genes or for making breeding progress. So real briefly, this isn't my area of expertise, but we used a modified digital genotyping version for doing marker development, aligned to B73 genome. And the one thing that's really unique is we only used SNPs that had at least 10x coverage, and we removed markers with more than 90% missing data. So there was very little missing data before we did the imputation, because we couldn't afford to misimpute something. So in total, we got 107,000 markers. The 2-way crosses each have less markers than that, because the parents have 5 minutes. And then there's about 11,000 markers that are polymorphic in both. Okay? And B73 oleic is very similar to the reference genome. The rest of these all are quite different between each other. So one of the things we see is these vast regions of identity by descent between these 2 parents, because they do have a common parent, about 25% that they share. But the other 2 parents, you can see the parentage switches quite often. So that's just a visualization of the parentage of the progeny, with each line across here being a different line. So if we use all this data to create our gold standard, we identify a log peak of 107 for Y1, or the yellow endosperm in maize. Okay? So that tells us, we have a lot of power to detect things in this population. And there's heterozygotes, missing allele calls, whatever. There's 1,100 observations there. Now we also did the color genes, and so the log peaks are less than that because it turns out colored 1 and R1 are epistatic, and you need both of them. So you don't have as much power in this population to find those things. And the red cob gene also shows up when we use a color here. Now what I want to get to is this. So this is the result of the various types of population development and how effective we are at mapping. So if we use all individuals, we can identify within 54 base pairs the Y1 gene. So very, very high resolution in this population. Super excited to see that. Even with the 4-way population with 3 generations of intermating, we still get the same resolution because the SNPs the limited number of SNPs was the issue there. Now as we go to these other populations, you can see that the confidence interval is much larger. In red here, if we only subsample 121 individuals, which is pretty common for a mapping population that people would use, okay, a linkage mapping population, we can see that in the 4-way 3 SIB, we would miss the peak quite a bit of the time, even with the confidence interval. Now, if we bump it up to 242 individuals being subsampled, we tend to be right on the peak. So that suggests for a single Mendelian trait, we need to probably be around 250 individuals for linkage mapping. I won't show you the other 4 genes, but I'll just show you paracarp color 1 here. We didn't have a lot of SNPs in this region, and so on the vast majority of populations, we actually missed the peak. Even by combining all of the data, we're still only within 88,000 base pairs of the QTL, but we still missed it. Okay, and that's a pretty scary thing, especially if you're going to devote a lot of resources to following up something. So I'll skip over the other two, the 2 blue color genes. It's a similar story. I mean, each gene is going to give you a different effect of resolution and accuracy in linkage mapping. But one of the things that we've really found is that a 4-way cross with intermating is going to be better for detecting loci, both accuracy and precision and effect, than the standard biparental population. But what really surprised me is the most effective thing, as Jesse Poland predicted, is increasing the number of individuals. That has a far greater effect than anything else. So small populations are not really that useful. So overall, I believe linkage populations are still really valuable, but I think we can improve the approach, and using multiple parents is a great way to do that. It only takes 2 genes to turn B73 into blue corn, and that was kind of exciting to see. And one of the goals of this project was really to map loci for recombination, but we can't make a genetic map. There's no software so far that can handle 107,000 markers on 1,200 individuals, and I see that as a major limiting factor. People think that linkage mapping is a solved problem, but I think we're really just at the beginning of trying to solve linkage mapping now. So we're going to use this population for a bunch of other traits, but seeing that I'm running out of time, I'll skip over my perennial corn and cycling gamete thing, and I'll go right to thanking the many students who have helped with these projects. You know, working with graduate students is one of the best parts of my job, and you know, they all have completely different projects and different personalities, and all those things have led to a lot of innovative insights. Most of our funding comes from Texas AgriLife Research, USDA AFRI projects, and then I get some money from the Texas corn producers to actually run the applied breeding program, as long as the hatch program support. A number of students have been supported on Monsanto Pioneer Fellowships or other type of fellowships. So with that, I'll leave up my acknowledgement and be happy to take any questions.